Welcome back to Chapter 22, Part 5. We're going to look at the role of time in the process of speciation. So, speciation, it turns out, can happen very rapidly or very slowly and can result from lots of changes or not too many changes. So everything's kind of in context. There are still lots of questions out there about how long it takes for a new species to form and how many genes it causes to happen and when, if a species are splitting in two, when they're actually considered to be new species. So let's just dive into some of that complication. Okay, so there are basically two main patterns that we see in the fossil record. And remember that we can't apply the biological species concept to the fossil record. So we use the morphological species concept, and we also use molecular data to help us figure out when one parent species evolves into two daughter species. And this can happen either quickly or slowly. Let's take a look at what happens if evolution happens fairly quickly. And again, in the fossil record, we see species that seem to stay exactly the same for a really long time. They just don't seem to have their appearance change at all. So, and then suddenly, for reasons that we may or may not know about, but suddenly, and this is the important thing, in the fossil record, it seems like they have changed. So here we see a species, and it seems to, over time, and here's our axis of time, right? It seems to fairly quickly go from being one species to two species. But then these two species stay relatively unchanged. This one's staying relatively unchanged, and this one's staying relatively unchanged for a pretty long time. So we call that punctuated equilibrium, or you might say the punctuated model. But and this, just so you know, punctuated equilibria is plural and punctuated equilibrium is single. So we could talk about many species going through punctuated equilibria, or we could talk about this species going through a period of punctuated equilibrium. Again, it's where it seems like a fairly rapid change from one species to another, followed by a relatively much longer time where they stay seemingly relatively unchanged. This is in comparison to the gradual model. Now, Darwin described the gradual model. We didn't know about the punctuated equilibrium model until 1970s. So, and remember, Darwin was writing in the 1850s. So quite a long time before we kind of saw a difference and had a scientific discussion about the difference between the rate of evolution. Until the 1970s, we pretty much just assumed the gradual model. And the gradual model shows, we see this in the fossil record, of one species changing a little bit, and then changing a little bit, and then changing a little bit, and another of the daughter species doing the same kind of change. This axis is still time, although it's not shown on here, but time is still the axis. So the gradual model says we have very slow and steady change over time from one species into another, and into another. So we have our two models, punctuated equilibrium and the gradual model. Is one of them wrong? No. There's not one that's wrong and one that's right. It depends on the evolutionary history of that species, because sometimes species can stay the same for a very long time, and sometimes they can have rapid divergence of new species. So both of those are shown in the fossil record. But we also see in labs that, in lab studies, that you can get pretty rapid speciation. The example here is Helianthus anomalus, and that is what happened in this, basically sunflowers, the species of sunflowers. And in a lab situation, we saw that this particular species of Helianthus and anomalus originated from the hybridization of two other species. That's very rapid, right? Happened in a lab situation. So that's pretty quick. That's like within a generation, right? There's exactly one generation, you get a new species. That's very rapid. But of course, the researchers didn't stop there. They wanted to look at what was actually happening at the level of the chromosomes. And so they started looking after several generations, and they saw that we were getting a lot of genetic change over a very short period of time. You can start to see here some divergences between the two chromosomes here and here. And so we've been able to experimentally show rapid evolution over short periods of time, as well as rely upon the fossil record to show us the slow and gradual evolutionary change that Darwin had talked to us about. If we look at some other research, we can see that there is just tons of information here about the speed of evolution. Scientists looked at 84 different groups of plants and animals and found that the interval between speciation events ranged from as little as 4,000 years in the fish and the cichlids, that's very fast, to 40 million years in beetles. The average time in these 84 groups that they studied, the average time between speciation events was 6.5 million years and was very rarely less than 500,000 years. So, right, this is pretty long, and but also highly variable, and that's the key here. There's no right or wrong speed at which evolution happens. We're just describing the things that we see. One really important question does remain, and that is how many genes influence the formation of new species? Again, I'm sorry to say that it depends, right? I don't have a solid answer for you. It depends on the species. It could be that just one single gene could make the difference, especially maybe if that gene is a gene that changes the time at which the organisms are mating. That could send them down a path of genetic divergence that they would never recover from. They just keep going down their separate paths. So one change can be a huge difference. Or in other species, it might take many, many genes in order for a new species to evolve. Remember the example of the snails that I talked to you about before. The left-handed snail and the right-handed snail, remember? That's the direction of the spiralness of the shell. It creates this mechanical barrier for mating. What I didn't tell you before is that the direction of the shell spiral is controlled by one gene. And so, of course, we're going to see very rapid evolution into new species if just this one gene has mutated. We can also look at this example of monkey flowers, which are called Mimulus. There are two genes that affect flower color in these species, two genes. And of course, flowers depend upon pollinators like bees and flies and butterflies and birds, right? Flowers depend upon pollinators to attract in and cause them to transfer pollen from one flower to another. Well, the color of the flower influences what pollinator is called in. Different butterflies prefer different colors, different birds prefer different colors. And so monkey flowers with different alleles at the flower locations, at the flower color locations, are less likely to interbreed because they are attracting different species of pollinators. Finally, we can look at this example in sunflowers. So in this species, in these various species of sunflowers, it's actually quite a large number of genes and gene interactions that help the isolation occur. So in the genus Helianthus, there are at least 26 sections of chromosomes, which you can kind of translate to the number of genes, sort of. We don't really know the number of genes, we just know the various sections of the genome. 26 of those sections are important in formation of the different species. So there's not just, sadly, there's not just like one rule that we can follow. There's a lot of different things that go into the evolution of different species. One thing I can tell you is that macroevolution is the large-scale evolutionary change, but it is due to the cumulative, the additive effect of many smaller scale processes and many different speciation events, and also many different extinction events. All of these go into play when looking at the large scale or macroevolutionary patterns that we see in the world around us. Well, it's been a really good semester. I hope that you have gotten as much out of this as possible. We've learned so much this semester, and it's been a pleasure helping you learn this material. If you ever have any questions, you can get in touch with me, and I look forward to hearing from you as always.